Everyone, my name is Dan Wegmann. I'm based at the University of Freiburg in Switzerland, and I'm here to kind of introduce you to the idea of incorporating genotyping uncertainty or genotype uncertainty in your models. Okay, so I'm gonna use different examples. They're mostly taken from population genetics, but I think like many of the ideas are easily kind of extended to other other fields. Okay, and I know that this is kind of a difficult endeavor for all of us, including myself. Like, we're all working on our drinks from last night and it's early in the morning. But so I thought I'd start with something super simple, like this is how sequencing works, right? I think like most of us are probably very familiar with this, but the idea is like we extract some DNA from cells. We have one or the other way of chopping this up into small pieces. We load this onto a sequencer and we get like short reads back, and like what short is depends on your application. And then we map those reads back against a reference, as we see up here. And now the challenge of genotyping stems from like the fact that we usually have a limited number of reads from that match one particular site in the genome. So for instance, consider this site here. It's like an AEC. And at this position, I have 5 reads. And it happens that 3 of them support an A, 1 a C, and 1 a G, because there's maybe a genotyping error or a sequencing error at that position. And so the challenge is now to try to infer the actual genotype from that data. Okay? And there's really 3 things that contribute to the difficulty. The first one is the limited amount of data. The second one is the sampling of alleles at the heterozygous site. There is a certain probability that we get only one allele covered and we miss the other one. And of course, sequencing errors. Okay? And because of all these challenges, we need usually quite a lot amount of data to accurately infer genotype. So this is just one way of looking at this. So these are some simulations we did for our own ancient DNA caller. And on the x-axis here, we have the sequencing depths. And that is the average depths in the genome. There is variation. And then we look at truly homozygous and truly heterozygous sites. And we just ask if we run our maximum likelihood caller, so that we'll call the genotype that has the highest likelihood, how often would we actually make a mistake? And the metric I'm using here is the fraction of alleles that are actually wrongly called, right? Like, so that is if a heterozygous site is called homozygous, then usually one allele is wrongly called, so it would be 0.5. But if you make a complete error, so you call a homozygous AA to a homozygous CC, you would have like a complete error, okay? But the key point here is that these error rates are actually relatively large, even if depth increases. So on the left here, I have the homozygous genotypes. You see we start at something like a little bit over 1%, but even if you go to 10x, you're still on the order of like 1%. Things are more drastic for heterozygous genotypes, where at 10x, we're always at 1%, always also around 1%, but you can down to like 10% error rates if you have really low depth data. Okay? And what that suggests is if your inference is based on actually knowing the genotype, you can invest a lot of money in sequencing your samples so that you actually cover each site sufficiently enough to be confident in your site. Right? Maybe just as a reminder, like, in humans, for instance, heterozygosity, so the fraction of heterozygous sites is about 1 in 1,000. So that means the error rate we're seeing here at a factor 10 is actually an order of magnitude higher than the true heterozygous sites you can have in your genome. So this, if you're interested in diversity, this is a real issue. Okay? So this is the point. However, we actually, I think, rarely care about the actual genotype. Most of the cases, we care about something completely different. Like in my example before, I'm caring about genetic diversity, like heterozygosity as the example. Yesterday, we see talks people caring about like local ancestry or like identifying parental segments. We care about demographic history. We want to maybe do GWAS. But all these things like are not the actual genotypes that matters, but it's more a hierarchical parameter. So for us, genotypes very often are basically a nuisance. Okay? But as I said just before, if we are now building our models around knowing the genotypes, for instance, in the case of diversity, we will drastically underestimate diversity if we have low depth. Okay? But if you think about this as a statistical problem, then how difficult can it really be to infer one number, like the fraction of sites that is heterozygous, from 3.2 billion data points in the genome, right? I mean, I would say that's hardly a crazy big data problem. We just need a way to actually account for the fact that we are unsure about each individual genotype. Okay? And that's the topic of the talk, and I think the general concept is like super simple, and then we're going to just see how we can extend different models to actually build that in. So that's the goal of today. So the general solution a lot of people are taking is to consider the genotype as a latent variable. Okay? So this is a DAG depicting a classic type of model we can imagine for these cases. So we have zeta up here, which is just a parameter or a vector of parameters of our model we're usually interested in. And you can imagine that we have maybe some probability distribution that is characterizing how we obtain genotypes under our model. So the model here could be a demographic, complicated model, and we want to know what's the probability of observing our vector of genotypes. And it could be much more simple. It could just be heterozygosity, the fraction of genotypes that are heterozygous, and have a probability distribution describing how I obtain the genotypes under this model. Okay, so this is usually what we care about from a biological point of view. However, we rarely observe genotypes directly. What we do is we obtain some sequencing data that is a function of these genotypes and perturbed with some errors here that in some cases we might know or some cases we might not know. Okay? And then if we write down the likelihood for such a model here, where we are interested in inferring our parameter of interest and potentially also the error model, we can write something like this. So we're interested in the probability of all the data we observe given our parameters of interest and the error model. And that turns out, in most cases, to be the product across all our samples that we consider independent in this regard. And then for each sample, we have to integrate out the uncertainty of the genotype. So we have a sum across the genotypes, and then we have the probability of the data, the observed sequencing data, given that particular genotype and the error model, times the probability of observing that particular genotype under our particular model. Okay? That's a classic way of integrating out a latent variable. Okay? And so this kind of formula that we see here, this model, is something that reappears over and over and over again if you want to incorporate genotyping uncertainty. Okay? And there's two parts to it. The first part is like the probability of the data given a genotype and an error model, and that's usually referred to as genotype likelihoods. And we're going to see in a second like a simple way of building those. And the other part, the second part here, the probability of the genotype given your parameters of interest, that's of course model specific. So that's where whatever model you use is coming in. So if you use a different model, then this is what you have to adjust. Okay. But anyway, like mostly we see equations of this form. Of course, it could be more complicated. You can embed that in a hidden Markov model or whatever, but this is, I think, the general form. There's the complication that we have here is this sum, right? For those of you familiar with inference algorithms, so we have a product here across something followed by a sum. So that just is a little bit annoying if you try to find, for instance, maximum likelihood estimates. And the classic solution to this is an EM algorithm, so an expectation maximization algorithm. And I mean, it's not for the talk of today. It's not super important whether or not you know that algorithm. I think it just matters that there is a numerical way of usually finding the maximum under these models. And for today, I'm just going to employ that algorithm for all our examples. And I'm going to finish off with a case that warns you that that may not always work, right? But basically, this is the setting we're in. OK, so briefly, genotype likelihoods. I think if you're considering building your own models with these, it's good to know that this is a general thing genotyping callers are actually producing. They're outputting genotype likelihoods under many, many different models. We can, for today, just think of a very, very simple model, which is, let's just say we have, OK, one of the cables. I'm not the first one struggling with this. I think I shouldn't touch not even a computer, but maybe not even a table much. OK, so basically, we have a very simple model in which there's only two alleles, and we can just say that every single read the sequencing machine produces is an independent kind of like sample from a particular site. So that is, we have here a product across all the individual reads, and we're just asking for all of them, what's the probability of observing that particular base that I see there as a function of the genotype. So I can build that genotype likelihood model. And the probability of observing a particular read here, we can have a very simple model that just says, if I have a homozygous genotype, say AA, and observe an A, I do that with probability 1 minus error, so I'm not making error. If I have a homozygous genotype AA, okay, and I observe something else than an A in my model with just two alleles, say a C, then I do that with probability epsilon, which is the error rate. And if I have a heterozygous site, I have like half. Now it starts to be a little annoying. Huh? So what should I do? I think it's one of the cables, but I don't know which one. OK, so if we have a heterozygous genotype, then I have, say, like it's AC, and I observe an A. I do that with a probability of not making an error with 1/2 because I'm sampling this allele, and I can also obtain an A by actually making an error and sampling the other allele, the C allele, right? If I have an AC heterozygous genotype, my read could be from a C, so I have a certain probability to observe an A. That's when I do an error, and I should not make an error in the other case, and it turns out it's just 1/2. It's a very, very simple kind of model. It's used a lot, but you can imagine that we can make that much more complicated. We can bring in the fact that there is 4 alleles actually, and not just 2. We can bring in haplotype structure if you want to have a more complicated caller that brings that into the genotype likelihood as well, right? But it doesn't really matter. The important thing is whatever tools we develop, we use usually these genotype likelihoods given by some caller. So you run your data through some caller, it gives you a big flat file with whatever type of genotype likelihoods you want, and then the idea is to use those in your own tools that account for the uncertainty in genotyping. Okay. And how to do that? Let me just make a super simple example. So for instance, we might be interested in inferring allele frequencies under a Hardy-Weinberg model. And so here we just have replicates across individuals. So for instance, we are interested in this allele frequency F. The data that I have is sequencing data from a bunch of individuals at these locations. I have an error model that is telling me how I obtain the sequencing data given the genotypes of the individuals. My genotypes are generated under a Hardy-Weinberg model, okay, which is just what I wrote down here. So we end up with a likelihood like this. And as I said, it's actually for this one super simple to derive an EM algorithm, and I can generate some estimates, okay? And if we run some super simple simulations in R, I just did that the other day for illustration purposes, we might end up with something like this. And I think that's actually quite telling, and it's telling us a lot about like what we should do in these type of models. So what I show you here is I show you the estimated allele frequency, the maximum likelihood allele frequency, as a function of sequencing depths for whether I have 20 individuals or 100 individuals, okay? And there's a lot of replicates that I did, and I assumed here that every single individual has always the exact same depth at this site. So a depth of 2 means every single individual has a depth of 2, okay? And what we see here is that basically in this case, adding sequencing depths to each individual is doing very little to actually reduce our uncertainty, our estimates, right? Like you see this here, I have 20 individuals, and there's quite some uncertainty associated with my estimate. So these are all individual estimates as a box plot. And as I increase steps, there's very little that is actually changing, okay? If you would zoom in and do more replicates, you would see that there's a tiny tick of reduction in like uncertainty, but it's very little, okay? If I increase to 100 individuals though, in comparison, like I have a much reduced uncertainty, okay? And what that tells us is that if I'm interested in estimating allele frequencies, what really matters is your sample size, not your sequencing, okay? And that's because the variation that you see is actually biological, not technical, right? My sequencing error rate is like 1%. I'm quite likely to see the correct allele. So every single read, if I have a depth of 1, a single read per individual, that read is most likely accurately telling me something about one allele in that individual. I'm unlikely making an error here, okay? But if I only sample like one individual, then I have an A or a C, and that's not telling me anything about the allele frequency, okay? If you want to get accurate estimates of allele frequencies, you need a lot of samples, okay? And so basically what that means is if we have means to take into account genotype uncertainty in our inference, it allows us also to actually use our money to generate biological replicates rather than sequencing replicates or technical replicates. I think that's a very general thing. Many people are arguing for this, and this is just a very simple example that illustrates this, okay? So if you're interested in diversity, what you should do is like just maximize the number of samples you can afford and minimize the amount of money you spend per sample, basically, okay? Now this is very good news for like a lot of data that I'm working with, because I'm working a lot with ancient DNA, and ancient DNA has some really interesting features, including it's extremely hard to actually get high depth data, right? And so in that sense, that's helping. So let me just show you next a few features of ancient DNA, and then some models we use to actually, or something we developed to estimate or characterize genetic diversity from ancient samples, taking a few things into account as the next example, okay? So what's special about ancient DNA, okay? Ancient DNA is data that is obtained from fossil bones. So these bones lay around in the ground for a while, like and for the samples that I have up here on the screen, it's about 10,000 years, okay? And there's a lot of degradation going on, and so there's very little DNA left in those bones. So if you were to touch those bones, there's more of your DNA in that thing than the person the bone belonged to, okay? So that's why people work in crazy suits and like in super clean labs in order to extract DNA. And just to illustrate how little DNA there actually is in there, what I show you here is a bunch of samples that we analyzed. And so what people do is there's usually two measures to kind of quantify how much you get out. The first one, how much individual reads you actually get out if you generate a library. So that would be here on the y-axis. And then how many of those reads actually are human, right? Because you have a lot of bacterial contamination. And that's like the endogenous DNA content that you see here, okay? So our best samples usually in the order of about 60%. So that means 40% of our sequencing effort is wasted because we sequence things that are not human at all. For some samples, that's much lower, like say below 20%. So it means 80% of the money we spend is not for the humans, it's not helping for the human sample. The other thing is, like for some samples, you just get like 100 times more DNA fragments out of what the people in those super clean lab consider super clean water, right? So these are like blanks where people just have like pure water in a like super high-tech facility, and so you still get some DNA out because there's contamination in the air. There's like always, like even though people try to keep it clean, and for many samples it's like just 100 times more DNA molecule than in a thing that is considered not to have DNA, right? So I would say there's very little DNA in ancient DNA samples usually. Okay, of course some are like exceptional, and then you have more, but it's a real problem. The second issue is that because of fragmentation of the DNA fragments that we sequence are usually very short, right? Okay, so these are some samples that were run like in the earlier days of when our collaborators were like super confident and said, yeah, 2 times 150, that's what I go for, and so they sequence 300 base pairs. But you see, like there's hardly, for some sample like this one here, there's essentially no read longer than 100 base pair anyways, and most reads are just like 40, 50 base pairs, okay? So that even though you get few reads, the reads are even short. Okay, and to make things worse, there is a thing called postmortem damage, which is particular damage that is affecting ancient DNA. It stems from the fact that if DNA is in the ground, there can be a deamination of like cytosins. They turn up to be uracils, and then if you sequence, have a PCR reaction on uracils, they become thymines, and so we see Ts, even though that person actually had a C. And we need to somewhat take that into account if you want to say anything about diversity, because the majority of like changes we see are actually not necessarily sequencing errors, but the sequencing machine is confident that it's a T, even though in reality it should be a C, because the change from C to T happened while the person was dead for a long time. Okay? And this is just showing you that the probability of having PMD is actually correlated with the position in the read, and that's very helpful, okay? Because that means we have a means of estimating and using or of estimating the rate of postmortem damage and building that into our model. Okay, so you may wonder, why is it? Sorry. Oh, you were why is it like position specific? Yeah, it's like because deaminations happen much more frequently if the DNA is single-stranded, and what happens to the DNA is like it breaks up into small chunks, and then you start to lose nucleotides at one end, so the ends are becoming single-stranded, and that's when they're no longer protected, and deaminations are happening more frequently. So that's why you have like, at least that's the theory. No, it happened after the person was dead and the DNA was laying in the ground for 10,000 years. So over that period, you have like the long kind of genomic DNA, and that starts to fragment, okay? So it starts to break up into small pieces, and at every end where you have like a break, it's quite easy to lose some nucleotides on one or the other strand, okay? So they are washed away, and then what you left is like with some single-stranded piece of DNA, and if you have a cytosine in there, and then there's like some UV light or something hitting, then there's a chance that you lose an amino group, and then it becomes a uracil, right? And then once we take the DNA out, like 5,000 years later, and sequence that uracil, we do a PCR reaction, the uracil becomes a T, and then the sequencing machine is totally confident that this is a T, because it is a T in the DNA, but it never was a T while that person was still living, right? So if you want to say something about the diversity of those samples, we have to factor those out, even though the sequencing machine is confident about it being a T, okay? And so genotype likelihoods now, and this is kind of like my advertisement here, genotype likelihoods are great because if your data has like particular types of features, it is actually relatively easy to extend those to take those into account. Like in our case, for instance, we have to build in this postmortem damage feature into genotype likelihoods. And there's 10 genotypes and like four bases you can observe. So there's like 40 terms. I'm not like boring you with all of those, so I just use like 2 as an example. But so this is what we can get. So imagine like the simple case in which we ask what's the probability that my sequencing read has an A, given that say my genotype is only T, right, from a haploid perspective, or TT homozygous, right? In this case, and an error rate. In this case, we say, well, the probability that you make an error is like epsilon. But then the probability that that error ends up being an A is like 1/3, because it could also be a C or a G, right? So we just end having epsilon third, okay? If now in the same case we have a same problem, but we have a genotype of either haploid C or like homozygous CC, and we ask what's the probability of observing a T, then we realize that there's 2 ways by which we can observe a T if we actually should have read a C. One is there could be postmortem damage, which changes the C into T, and then my sequencing machine is not making an error at all because it's confident that this is a T, and it's right, it is a T in the DNA that I give the machine, right? So that would be the first term. So I have D, the probability of making a postmortem damage at this position, and not making a sequencing error, or I can actually not have postmortem damage there, so there's still a C when I give the DNA to the machine, but the machine is then making a sequencing error, right? So it's quite easy to extend, and I can do that for all kind of combinations of genotypes and data, and I can easily extend the genotype likelihood model that take into account a particular feature of my data. Like if other data has other particular features, it's usually equally easy to do that, okay? Should also say, because of these exponential decay patterns, it's actually quite easy to learn the rate of PMD from the data, and I'm not going into that, but like the basic trick is to realize that if you compare the reference to a sample, there's some tree connecting these two, and there's mutations happening on that tree. But if these are real mutations, then whether the reference is C and the sample T, or your sample is T and the reference is C, there's no reason for that not to be different, right? Because it happens, it's symmetric on the tree. And so we can use the T to C rate to inform us about real divergence between, say, the reference and my data, and like the excess C to T that we see is the one we need to explain as PMD. Like the models are a bit more complicated, but that's the bottom line. Okay? So now that we have like a genotype likelihood model for ancient DNA, we can ask, so let's have a model to infer genetic diversity, okay? And so we decided to use a very simple model that was proposed by Felsenstein in 1981. And so the model here is very simple. It assumes that there are two sets of parameters. One is base frequencies in the genome, just like what's the probability that a random base is an A, C, G, or a T, and we call pi. And then a substitution rate, okay? And so the idea is like we have this tree here connecting my two chromosomes in the same sample. So we have one site in one individual, and I have two alleles, and then these are connected by some tree, okay? And what I'm now asking is simply like, what is the probability that I observe like one base at one end of the tree and another base at the other end of the tree, given like a probability of a distribution of bases in general and some substitution rate along that tree? Okay. And so it's maybe quite easy to see in the latter case. So the probability if these two bases are actually different is just you need to have at least one change along that tree. You can have multiple changes, multiple substitutions, but there need to be at least one substitution, which is given by 1 minus e at the power of minus my rate. And if I do have a substitution, I have a certain probability that on the left side of my tree I start with allele k, that's given by pi k, and that the last substitution that I have in my sequence turns up being an L. So we just say that every substitution picks a random base from my distribution, so it's pi L. Okay? If it's homozygous, then you have like these two things. There could be no substitution, or you can have substitutions, but the last one picks again the same allele that you started with. So that's the basic idea of Felsenstein. Okay? And so we have now these two sets of parameters. We have pi and zeta, and we'd like to estimate those. And we can build a likelihood just exactly of the form we've been discussing so far. So I want to integrate out genotype uncertainty. I have no clue about those genotypes. I build in here my genotype likelihoods. Okay, and I bring in PMD at this location, or at this probability, and I have this Felsenstein probability here in the back. And now I want to estimate zeta. Okay, and yeah, let's do some simulations to see if that actually works. And it's maybe not that surprising, but we don't need a crazy amount of data to infer such a simple thing as just a parameter, like one single parameter like zeta. So what you see here is the estimated zeta, where the true one is 10 to the minus 3, which is like roughly where we expect humans to be, as a function of sequencing depth. And you see again like that as depth is a bit above 1, we have like very, very good estimates of this. Okay. We can also, this was for like a window size of 1 megabase, so we're using 1 million base pairs. What's interesting is you can also compensate lack of depth with more sites, because more sites also give you more information. So if we fix the depth to 1x, but we have like very small to very large windows here, so we're using more or less sites, using more sites also improves this accuracy. And it turns out these two are actually completely complementary. So here's just showing a distribution, or like an error rates as a function of both window size and depth. And you see you can always increase one or the other, and it can really compensate each other. So that's kind of a neat trick of accounting for uncertainty. If you're very uncertain at one point, just use more points, and then you also get like information if it's expensive to get more information per point, right? So for instance, if we have genomes that have a crazy low depth, then maybe we just say we estimate a diversity on the whole genome, because then we have much more power to do so. And if we have a high depth genome, say, oh, maybe we can zoom in onto 10 or 100 kb windows, because you have enough power to estimate zeta there. Okay. And so when we developed this, we were like super excited and said like, yeah, let's run this on our Greek samples. And then we did that in like 1 megabase windows. And these are just 3 samples. There's an early plot. My student sent me, totally exciting. Hey, it works, you know, like everything is fantastic. But then you just look at this, you realize like it actually doesn't work at all, right? Because you have these 3 samples, they're all human. They should kind of like be somewhat similar. They're all about 10,000 years old in the same like region. And what we have is we have some individuals like this black one that's maybe 10 to the minus 4. You would say, oh, like a little low diversity, but it's maybe possible. But then you have this guy here that is essentially every other base pair or so is like is heterozygous, and that's like very unrealistic. And then you have this other guy that's a weird feature, so it's either like 10 to the minus 10 in some windows, so absolutely nothing is diverse, or like 10 to the minus 5. And so there was this obvious question, is like, why are these estimates so different, right? Like, I mean, basically we have this fancy model, it uses all these data, and we want to estimate diversity. We just said it's actually a simple problem, but somehow it completely fails, okay? And the answer is, and that's really a big lesson learned, the answer is like, if we want to take into account uncertainty, we have this error model that tells us what's the probability of actually observing the data given a genotype. But if this error model is, of course, not accurate, then like our estimates are not accurate, right? We rely on this error model. Okay? And for most part, what people do is like we rely on the quality scores that are given to us by the Illumina sequencing machine. If the Illumina sequencing machine tells me it's quality 20, I translate this to an error of like 1%. Okay? And I just trust it, or that's what we did here. We trusted those quality scores. But it turns out like these quality scores are not trustworthy at all, right? If the Illumina machine tells you, oh, I make a 1% error, then that does not necessarily mean that the machine makes a 1% error. And if you don't correct for that, but there's variation in like how wrong the machine is, then this translates directly into differences in diversity, right? So if the machine is like overconfident for one sample, then we can increase diversity, because whenever we see 2 different alleles, we say, yeah, that's like a heterozygous site. And if my machine is underconfident, then whenever we see differences, say, oh, that's probably a sequencing error, and I underestimate diversity. Okay. So somehow we have to deal with this, and I think we have to deal with this in all cases in which we use genotype likelihoods. It's like a very important part. So we thought about how could we actually learn like about these issues. And so there's tools out there, like the most famous one is probably BQSR, which is implemented in GATK, but the basic idea they use there is you use a reference genome and you use all sites that you know are polymorphic. So you blank those out, and then you just look at the other sites and pretend you know the allele. It must be a reference allele, okay? Now, if you work with modern humans, that may be a very sensible approach. Like, it may also be with ancient humans. We were a little scary of using modern knowledge to actually infer like recalibration parameters for ancients. Like we were afraid of making them look too modern. But also, if you work with non-model organisms, you may not have like this great knowledge about reference genome and so forth. And we saw a little bit how can we maybe learn these error rates without actually making use of a reference genome and known polymorphic data. Okay, and the first idea that came to our mind was this one here, which is that like, well, there's always parts of the genome where we actually are pretty sure there's no polymorphism, right? So these are pseudohaploid or haploid regions in the genome, such as, for instance, mtDNA, or if you have male samples, it's X-linked data, right? There should be no polymorphism on the male X. Or if there's like ultra-conserved elements that are like completely conserved across all vertebrates, so it's unlikely that your sample is heterozygous there. And so for these regions, we may not know the actual base, right? I may not know what is there, but the only thing I do know is that if I see 2 separate bases, 2 different alleles, one of them is wrong, right? And I can try to exploit that information to kind of learn an error model, okay? And so this is what we did, and you see, again, the way to do this ends up with this almost exactly identical equation where we have the probability of observing the data given some genotype, and here's some error model. I have the probability of the genotype given some distribution, like here it's pseudohaploid, so I just use the distribution of bases in the genome. And now this epsilon here is what I want to learn. Okay? And again, I can use an EM algorithm to do that. Okay? Error rates are bounded between 0 and 1. So we employ the logistic model and like had just informing the logistic model using a linear combination of like other things you know about this space, right? The quality score that the machine tells me is informative about it, but also maybe the position in the read, maybe like other things like just the base pair that was read before, like we call this context. So all these things you can build into your model. Okay? So we did that for our samples, and that's basically the pattern that we do obtain. So what I show you here is the kind of a density plot of the machine-reported quality score and the one that we learned, like the transformation of the one that we inferred. Okay? And we've now done this on a lot of samples, and I can tell you it almost always looks like this. Not always, but almost always. And so what the message is like Illumina is overestimating its own quality by quite some margin. Okay? So for instance, if the Illumina tells me it's a quality of 40, it's more likely a quality of 30, right? So my error rate is 10 times higher than like what the machine pretends, okay? And that's not always the case, but it's like these patterns, so these are 3 samples, and you see how similar they are, and there's like 50 more, and they all look almost the same. So it seems to be a very common thing that like Illumina is somewhat overestimating its own quality in most cases, okay? It's a little sad because like if you go on their website and there's a new sequencer and say, it's fantastic because we produce 80% of our reads at quality 20. Yeah, well, if you choose the quality yourself, that's quite an easy thing to reach, right? Like the question is whether it's actually real, this quality score, and it doesn't seem to be. Okay, yeah, sorry.
I was going to ask on a previous slide, do you have an explanation for why on that leftmost, the next one?
On this one?
Yeah, on the leftmost plot, why at like 38 or so, you actually have a binomial?
Yeah. Yeah, I have like, so basically, there's multiple things contributing to all of this. Like the first one is there's different regroup. So usually if you have in ancient DNA, people actually have to do multiple sequencing runs to get their data. And so all of them have different parameters, but they're mixed into one plot. And the newer Illumina machines, they write FASTQ files that are smaller, so you can zip them by actually binning the quality scores. So they only use, you see like these bars here. Okay, so these are like some of the runs were done on machines that would only give you like binned quality scores. Okay, so they don't give you the full spectrum anymore because then the zipping of the files is more efficient if you have less difference in characters. Yeah. It doesn't. If we did simulations on this, because I thought like this is when they brought this up, I thought this is crazy, right? We lose a lot of information. But so if we artificially bin and then like check our performance, it truly has a very limited effect. It's kind of interesting. I thought it would be drastic. Yes. Library by actually sequencing, library by library and sequencing run by sequencing run. Like because the machine, they read their adapters or something in the beginning, and then based on this they try to fit an error model, and that's then applied to the run. And so all the runs are like different. You have to recalibrate them differently. But there's also some library prep issues that can affect your error rates, and so we also do it for library. Yeah. So then we have like exactly. That's like for one sample, like the whole data pushed together. That's what it is. Yeah. Yes, I mean, yes and no, right? Like, so basically, this is a great idea, but what people started doing is sequencing single end because of the overlap, right? Like, so basically there's so much overlap usually because the base pair, there is only 40 base pairs or so, that like you waste so much money that people stop doing like paired end sequencing. And so what we did then is like we decided to do something else, which is we went through all the paired end data, and then when there was overlap, we would actually use them as independent samples in our model because we are interested in the machine errors. And for the machine errors, there may be like they are independent. Machine is doing 2 times an error like completely independently, right? So you can actually use them as completely 2 separate data points in this model. And then after recalibration, we merge the reads. So we actually take only the base pair is the higher quality. But for this model, you don't even have to, you can just use them as 2 data points in the model. So in that sense, we're exploiting it. Because we are interested in what the machine makes, right? It's not the PCR errors. Yes? Absolutely.
There was a community that they could follow the samples in PCR reaction by live using random adapters, and 30% of these patches were coming from PCR errors. And they also showed that you could eliminate PCR errors by eliminating also money with your library preparation. But there is one more thing you cannot deal with: DNA damage during DNA extraction.
Yeah.
So I'm always thinking, I'm very interested in what you can do with DNA now, but DNA damage uses a lot of errors, especially damage DNA in the environment during DNA extraction, and we need to use different enzymes to avoid that.
Yes.
It's not very feasible. So there are all these problems that we're not going to be able to resolve. Still, we're seeing anything, so I think that's all fine. But
yeah.
I'm not the only problem.
Okay, so that's a very, very fair point, and actually like one of the next examples that are coming up are actually talking exactly about PCR errors. But like, let me just say this, like the cool thing I think about some of these recalibration tools, like not only ours, but for instance this one, is that we're actually just learning the rate at which there's mismatches in places where there shouldn't be mismatches, right? Like if I take a male sample and go on the X chromosome, there shouldn't be a mismatch. And I'm just learning the probability that there is a mismatch as my error, okay? And if that error rate is now a sequencing error rate or contamination or whatever it is there, actually my model doesn't care. It just tells you at this pace, at this position, all reads that I have should give me the same base. If some of them don't, I call this an error. Like I would argue that some of these recalibration methods actually take into account a lot of these errors by doing that. The danger is, for instance, if you do ultra-conserved elements as the recalibration, you will not take into account contamination because all the other humans are the same there. But if you, like there is a Kai Prüfer, Max Planck, he has like this other, it's called SNP-AD, and like we're not thinking of like merging the ideas, but he uses like a slightly different model that is not relying on like regions in the genome that are pseudohaploid. And so he can even take, I think he in factors therefore in contamination as well. So I think like you're absolutely right, PCR errors constitute a large problem for this. But if you have means of just estimating rates at which you have mismatches even though you shouldn't, I would argue this rate actually reflects this. And it might, well, if I wanted to say something nice about the Illumina, it might well be that a large part of this like actually stems from PCR errors, right? I don't know. It's just like, I think the important thing is for me, like if in my BAM file I have a quality of 20, that does not tell me something about the real mismatch rate, right? Without saying where these mismatches are originating from. Yeah. Yes? Oh, I have only 4 minutes left. Oh my God, okay. So, okay, so now I have to think what to do. Okay, actually it's cool data and it works on ancient stuff and like I think you don't care about data anyways, okay? But like there's maybe, now I have to think what I do. Like there's, I'm gonna spend 2 minutes on 2 topics just to raise some issues. So like the first one is like bloody RAD and it goes really into like the thing we've just been discussing. So I had, maybe you know about RAD sequencing. It's like this cheap way of generating data across the whole genome by exploiting like restriction sites. So you actually use resistant digestion and that's where you add your adapters and then you only sequence these parts. And so the trick of this is that you're only going to sequence like a fraction of the genome, but you can sequence the hopefully same fraction of that genome in all your samples that you have. And you're exploiting restriction sites to do that, so you don't need a reference genome or anything to do that, right? So it sounds like super cool. The problem is just there's lots of PCR steps involved in RAD. Okay, and so therefore there's lots of known issues. And I had one student that had like RAD data, and it looked like super crappy. And so we were interested in like maybe having a way to quantify and correcting some of these errors. Okay, and so luckily she had some replicates, and that's maybe something I would advise people to do anyways. Just if you do 100 samples, like do two times, right? It's not costing you a lot, but having replicates on the same individual gives you a handle on learning a lot of issues in your data by comparing them. And so she developed this method here, which would essentially just where we assume that we have multiple observed genotypes from the same individual, and there is a true genotype that we don't know, that is a latent variable, and we want to kind of integrate over that while learning an error model for this, okay? We had a very simple error model that just said we have a per allele error that depending on whether the true site was heterozygous or the true site was homozygous. Like super simple, okay? I can explain you the details later. It's really just I want to make this point that, so these are the results we got when we applied this to her replicates. So she had two data sets, like the black one and the red one, and like the filled symbols here are the error rates for the heterozygous case, and the open one for the homozygous case. And you see that if you have RAD, in our case at least, with a depth of 30, you would still have like several % error rates like on your genotypes, right? And then on the heterozygous ones, like sometimes even tens of %, right? And so that's kind of crazy. So it also shows that with increasing depths, we're not correcting this at all, right? And one of the ideas is that this is particularly because of PCRs in this case. But the cool thing is, if you know these error rates, you can actually update your genotype likelihoods, right? I can build a new likelihood with it, the probability that I call genotype G, given that the true genotype is like gamma, right? And I can use this as my genotype likelihoods. And when she does this, so she had like crosses from like hybrid species, and she was doing chromosome painting. And what you see on the left is this is supposedly a backcross, and gray here are heterozygous ancestry segments, and blue is from one of the two species. And on the left is the raw data she got, and she definitely didn't see a backcross because there were all these orange blocks, which would suggest that the ancestry is homozygous for the other species, right? Even though you know it's a backcross. Okay, so you have two species. You generate an F1. The F1 is backcross to one of the species. Okay, and now in the genome, I'm trying to infer what is the ancestry in every variant along the chromosomes. And the ancestry can be one of three types. It can be homozygous, the first species. It can be heterozygous for ancestry, homozygous, the other species. And if you have a backcross, you should see heterozygous and homozygous segments, but never homozygous of the other species. So all those orange blocks here, they shouldn't exist because we know it's a backcross. Okay? But this is the RAD data we use. Now, if you actually update genotype likelihoods, like by using this correction model that I did, and then she runs it, like there's still some small issues, but we largely correct for this fact. But we have to do it, and increasing sequencing depths does not solve the problem, right? So this is the message, right? Like thinking a little bit about the errors in there may even be important if you have high depths, if you run a protocol that have data that has particular features, okay, such as high PCR errors, okay? And yeah, maybe the last thing I kind of do, like just have 1 minute left, but I just want to show you like this result here. So basically, you can apply the very same principle of accounting for genotype uncertainty, as I said, to a lot of different models, right? Including like genotype, phenotype association models, okay, where it's actually quite simple. Show you the DAG, you just say like, okay, I have a genotype that is affecting my phenotype, but it's also generating or is affecting some sequencing data that I get from it. So I have to integrate out the genotype, I don't know it. So you can build a model, and we just, for the fun of it, we did that. And I run here some simulations where I assume the following. So you have a total budget that would be, say, enough for 1,000 to sequence 1,000 samples at 20x. Okay? And I assume that library costs are like $18 and sequencing costs $36 per 1x. That's roughly what I have to pay my sequencing facility, okay, on the NovaSeq. And then I just asked, so I have a fixed budget, and I can either sequence a lot of individuals low depths, or I can sequence few individuals at high depths for my budget, and I have this choice that I can do. And I wanted to know, so what's the best strategy I should do? And that obviously potentially depends on like how strong, like I want to find sites in the genome that are associated with my phenotype, and depending on the explained variance of that I have at this site, like I might use a different strategy. So I simulated like an explained variance of 0.1, so that means like this particular site explains a lot about the genotype, really a lot. Okay, or something where it explains very little and something intermediate. Now, obviously, if it explains a lot, no matter what you do, you're going to find it. Okay, if your site, so your genotype at these locations, they don't explain hardly anything about phenotype, no matter what you do, you won't find it. Okay, but the cool thing is, like, if you have something intermediate where you actually have the power, and you see here as the power to detect those sites in my simulations as a function of depth, you should definitely go for low depth sequencing in a lot of samples. Like your power is way, way, way larger. Okay, and again, that is because the real noise in your GWAS stems actually from the fact that the phenotype is noisy and the genotypes are very variable across individuals. And it doesn't stem from like sequencing errors, which are just 1% or something like on an individual sequence level, right? So my take home therefore is like if you build in ways to account for genotype uncertainty in your models, you can actually manage to lower costs or improve like the amount of information you can extract from data, simply because you can take into account biological variation rather than technical variation. Okay, so that's my first take home, and I think the other one, as I said many times, is how can you turn noisy data into biological insights. So basically, model error rates explicitly. If you know something about them, build that in those models. Like in ancient DNA, you know about PMD, you may know about other features in your data, and then infer hierarchical parameters by integrating out the uncertainty as a concept. That's very promising. Okay, so there is we have a Bitbucket page with all the code. There's like 3 programs, Atlas, Sponge, and Tiger, to implement the things I've been talking about. And also, I would just want to say that we just have an upcoming chapter in the new handbook of statistical genetics that I think is going through a lot of these ideas and examples with all the derivations in case you want to follow up on those. Okay, thank you. Yes, John. You mean like the orange blocks? Yeah.
Like the errors would be sort of sprinkled and not clustered. But I don't know the structure of RAD.
Yeah. No, I think you're right. Like that's what you would expect. I think my feeling is it's more a feature of chromosome painting, because in order to actually to estimate a particular type of painting, you actually need multiple sites like contributing to this. But then you actually tolerate errors too, right? Because there are errors. Like their genotyping errors are kind of built in, so you allow for errors. So even if the question is just in some region, which of the genotypes is the most common, right? Like, and so that just means you identify the regions where maybe by chance these are more common. It doesn't mean that all of the sites have this area, of course, right? It's just like they're more common, while in other regions the other ones are more common. So I think if you have just these sporadic errors, we would never see them because the HMM would actually just correct for that, right? So we only see the regions that are enriched for these sites. But if you imagine, like in this case, we had a genotyping error rates on heterozygous sites above 50%, right? Above 50%. So half of the heterozygous sites we call homozygous, right? So given that, we actually see maybe few like orange blocks, right? And see how well the HMM kind of works. But yeah, it's like drastic in that data. Yeah, Bogdan.
It's really exciting that you can get all these I'm just curious whether you give some thought about looking at other types of techniques, for example, ChIP-seq and so on, whether there's a similar type of trade-off with depth versus sample.
I have personally not really worked on this type of data, but I can imagine that I just like right now, I think RNA-seq would be a good example where there's actually quite some noise in estimating the real expression rate. And it's even more complicated because you have a limited number of reads you generate. And so if you have one gene that is like very, very highly expressed, then it means that the reads, the amount per million reads that you get for other genes is lowered, right? Like simply because there's competition. And so this is computational data, and I think for many genes this is very low. So if you had a way of having many replicates of individuals, and then you want to estimate, say, changes between two sets in expression, but you would account for the uncertainty in actually estimating expression rates per sample, you should improve your power, right? And you can even bring in the compositional aspect of this in your model. So I would just guess, like, in theory, it should help, like, in a lot of fields, but of course one would have to develop, like, different likelihood models for all of these things. But I would say, like, it might work very well. But to the degree to which it's worth it, like, financially, I don't really have a good feeling, right? I just think it turns out that when you are interested in diversity and sequencing, it's worth it because, like, you really usually are limited in the sample size because of money. And just by investing your money on more samples rather than more sequencing usually helps, right? But whether that's true in an RNA-seq study, I have no gut feeling about that. Yeah.